Okay. Assalamu alaikum warahmatullahi wabarakatuh, everyone. I'm Abdulrahman Farahat. I'm a second year medical student, and our lecture today is going to be about metabolic effects of insulin and glucagon. It's a really simple lecture, and I already spoke a lot about insulin and glucagon in the endorepro blog. And I'm going to touch upon certain things. My main objective is to focus on the stuff that you guys might get asked on. I might go a little quickly through these slides. And yeah, so if you want to contact me, here are my email and my phone number, and let's begin. Okay, so first, looking at the table of contents, we're going to discuss insulin, and then we're going to take a look at glucagon, and finally, we're going to speak a bit about hypoglycemia. Now, for insulin, as you all know, insulin comes from the islets of Langerhans within the pancreas, from the beta cells themselves. And now, the effects of insulin are essentially anabolic effects. So when we say anabolic, we mean that they are building upon. So we have a molecule, we're building up molecules, and this would involve synthesis of glycogen, triacylglycerols, or proteins. Now, as you know, speaking about the structure of insulin itself, it's made of two chains, an alpha chain and a beta chain. The alpha chain consists of 21 amino acids and the beta chain of 30 amino acids, and together they are 51 amino acids. There are intramolecular disulfide bridges, and between to connect both chains together, you would have two disulfide bridges. And for the synthesis of insulin, you would start with pre-proinsulin, and then we cleave the signal sequence. We'll go into the proinsulin, and by cleaving the C-peptide, we'll get insulin and the C-peptide. Does anybody know what's the importance of C-peptide? Okay, so C-peptide essentially has two main functions. Okay, C-peptide has two main functions. So it is essential for proper insulin folding. However, this is not as clinically relevant to us. The important thing about C-peptide is that it is a good indicator for insulin production. And what do I mean by that? If you imagine a diabetic patient, and this diabetic patient is already taking exogenous insulin, so he's taking insulin from outside to control his diabetes, to control his blood sugar. Now, this exogenous insulin does not have C-peptide with it. And a way to measure whether the insulin within this patient is his endogenous insulin or exogenous insulin is by looking at the C-peptide levels. So a C-peptide level would indicate that this patient is producing insulin. Now, the final thing we're going to take about this objective is the degradation of insulin. And so, degradation of insulin. And as we all, yeah, it's fine. Okay. For the degradation of insulin, we have so insulin has a short half-life of 6 minutes, and this is important for us because insulin is very acute in its action, and it has a very rapid response when it comes to controlling the levels of blood glucose. Now, moving on to the effects of glucose, protein, and lipids on the secretion of insulin itself. So how does glucose, protein, and lipids cause our body to release insulin? So we'll start with glucose. As you can see from this diagram right here, glucose, you'd intake glucose, it would get absorbed from the GI tract and into the blood. In the blood, it reaches the islets of Langerhans within the pancreas and the beta cells specifically. The beta cells have this specific transporter called GLUT2 transporter. I meant to write it here, but I forgot. So it's a GLUT2 transporter that brings in glucose. This glucose then gets phosphorylated, and so we trap this glucose, and it becomes glucose 6-phosphate. And as you know, glucose would enter, it would go through glycolysis, Krebs cycle, and so on and so forth to produce ATP. Now, this ATP is important because it inhibits this potassium-sensitive ATPase. By inhibiting this potassium-sensitive ATPase, the potassium will not be excreted outside the cell, and this would cause depolarization of the cell itself. This depolarization is going to stimulate the calcium channels, and so calcium channels will then enter. Once calcium channels enter, these insulin-containing vesicles would fuse with the cell membrane and release their insulin. Am I clear if I'm going too fast? Okay. Now, an important thing to keep in mind is that now imagine a type 2 diabetic patient. This type 2 diabetic patient, we usually give them drugs to help them release their insulin. And so this drug acts by blocking this potassium sensitive ATPase. And this potassium sensitive, again, potassium would not get released. There would be depolarization within the cell itself, and calcium would enter. When calcium enters, this vesicles would fuse with the cell membrane and release their insulin, right? Okay, so does anybody know what is the name of the drug that we give? Can't see the chat. Yeah. So that drug would be sulfonylurea. So I want you guys to keep that in mind because sulfonylureas is a pretty high yield concept. And so essentially it's a drug that binds to the potassium sensitivity ATPase and prevents the release of potassium. And so depolarization would happen, calcium would enter, and insulin would be released. Now, we spoke already about how glucose would lead to the release of insulin. Now, moving on to the amino acids and the fatty acids that would lead to the release of insulin. And so essentially, amino acids and fatty acids would help glucose in releasing insulin itself. And so it's not a really direct effect. Okay, so we're done with this objective as well. Is this clear with everyone? Okay, so now to the gastrointestinal peptide hormones. So we have gastric inhibitory polypeptide and a glucagon-like peptide. And their main action is that they increase the sensitivity of beta cells to glucose. And so by increasing the sensitivity of beta cells to glucose, you are increasing essentially the insulin release. And so how that happens is you take something orally and it goes through the GI tract into the small intestine. And within the small intestine, these hormones are going to get released, the gastric inhibitory peptide and glucagon-like peptide 1. And as we said, they increase the sensitivity of beta cells to glucose. And so these beta cells will be more sensitive now and they release even more insulin. And that's why it's important to note that if we want to increase the amount of insulin release, we're going to give methylene glucose orally instead of giving it IV. And that's because of the gastric inhibitory peptide and the glucagon-like peptide 1. Now, we spoke about how insulin gets released and how the gastric inhibitory peptide and the gastrointestinal peptide, essentially hormones, work to release insulin. Now we're going to speak about something that inhibits the release of insulin. And so if you know this from Winnie the Pooh, Piglet is a very anxious character, and he's always in this tense state. And so he has levels of norepinephrine and epinephrine. And these high levels are essentially going to do two main things. They're going to mobilize your fuse. And so essentially, you're going to break down your glycogen storage. You're going to build up more glucose in the blood. And you're breaking down fatty acids. There will be fatty acid oxidation to provide energy. But the important thing I want to stress about would be the inhibition of insulin secretion. So norepinephrine and epinephrine inhibit insulin secretion. Okay, now we're done with gastrointestinal peptide hormones and everything else. On to the last thing regarding insulin, it's going to be effects of insulin on glucose, protein, and lipids. For glucose, there will be three main things. Increased glucose uptake, and this happens because insulin activates GLUT4 transporters. And GLUT4 transporters are found in the muscle and the adipose tissue. And so you're going to increase the glucose uptake from the blood. The second thing is you increase glycogen synthesis. And as we said before, insulin is an anabolic hormone. And so we're going to build up more molecules, and that's through glycogen. And finally, decrease production of glucose. Because again, your main objective with releasing insulin is to reduce the amount of glucose in the blood. And so by giving insulin, this would reduce the production of glucose because it inhibits glycogenolysis and gluconeogenesis. Okay. When it comes to lipids, again, there would be increased glucose uptake. And you might ask how is glucose related to lipids? Well, again, that's important because we said glucose, there's increased glucose uptake in both muscles and adipose tissue through GLUT4 transporters, right? Now, the glucose that's taken up into adipose tissue is important for triacylglycerol synthesis because this glucose provides a certain molecule called glycerol 3-phosphate. It's important for triacylglycerol synthesis. And again, that's an anabolic reaction. You're building up. It also activates lipoprotein lipase. And so lipoprotein lipase essentially at the end of its pathway, there will be increased triacylglycerol synthesis. And the last thing would be that there is this dephosphorylation of the hormone-sensitive lipase, which is an enzyme inside the cells that breaks down fatty acids. So it's increasing the amount of fatty acids. And you don't want that. You want anabolism, not catabolism. Now, for proteins, again, it's really simple. There will be increased take-up of amino acids, and there will be protein synthesis. Okay, now we're done with insulin. Does anybody have any issues with insulin? Is anything not clear? Okay, moving on to glucagon. So when it comes to glucagon, again, it's a hormone released by the alpha cells of the pancreatic islets of Langerhans. And if you know glucagon, it's important to maintain or increase the level of glucose in the blood in cases of methane fasting or others. Now, for the structure of glucagon, as you can see, this is a peptide that consists of 29 amino acids. Again, like insulin, it starts as pre-pro-glucagon, and then it divides into pro-glucagon, and finally glucagon. And the last thing is that it is a catabolic hormone. We said insulin was anabolic, so you're building up. Glucagon would be a catabolic hormone, and so you're breaking down through glycogenolysis, or you could also build up through gluconeogenesis. But it's mainly a catabolic hormone because it breaks down glucose, breaks down glycogenolysis, glycogen, it breaks down lipids, triacylglycerols into free fatty acids, and it breaks down proteins. Now, when is the glucagon released? So glucagon would be released. Okay, so when is glucagon released? Glucagon will be released in three main scenarios. One, when there is a decreased plasma glucose, and that's our main stimulant. So when there is hypoglycemia, the second there is hypoglycemia, glucagon will be released. The second thing is that it is an when there is amino acids from a protein meal, and this would also be a trigger for glucagon to get released. And finally, the thing is, the most important thing would be catecholamine release. So imagine, why would catecholamines be released? They're usually released when there are stressful situations. And so in stressful situations, you're going to need to break down and provide energy for the body's tissues. And so that's why when there is a lot of norepinephrine and epinephrine in the blood, glucagon is also released along with them to maintain the glucose levels in the blood and provide energy for the body. Okay, now onto the metabolic effects of glucagon. So again, we're going to take a look at its effects on carbohydrates, its effects on lipids, and on proteins. When it comes to carbohydrates, glucagon, as you said, it wants to increase the levels of glucose in the blood, right? So it's going to do two main things. It's going to increase glycogenolysis, or breaking down glycogen into glucose, and it's going to increase gluconeogenesis. And so you're essentially building up glucose from it. Now, for the lipids, it's going, as you said, it's going to break down lipids into free fatty acids, and there will be formation of ketone bodies. For proteins, there will be increased uptake of amino acids, and if you know from another lecture about protein synthesis, certain amino acids are gluconeogenic, and so what that means is that they could enter into Krebs cycle and essentially go back all the way and helping gluconeogenesis. But that's the whole purpose of increasing uptake of these amino acids. Now, For the mechanism of action of glucagon, again, it's really simple. If you take a look at this diagram right here, you can see this glucagon molecule is going to bind to the receptor at the cell membrane right here. And this receptor is going to activate adenyl cyclase. Adenyl cyclase is going to change ATP into cyclic AMP. Cyclic AMP is going to stimulate protein kinase A. Protein kinase A is going to phosphorylate multiple enzymes. So it's going to phosphorylate multiple enzymes, and these multiple enzymes are going to help in increasing gluconeogenesis, glycogenolysis. There will be fatty acid oxidation, ketogenesis, uptake of amino acids. So essentially everything we already mentioned here. Yeah, everything we already mentioned here. And in addition, there will be decreased glycogenolysis. Oh, this is wrong. There wouldn't be decreased glycogen. It should be glycogen synthesis. Yeah. So there would be decreased glycogen synthesis, not loss. Okay. Now, for hypoglycemia, our last objective. For hypoglycemia, as we know, there are two main systems that regulate glucose during hypoglycemia. The glucagon pathway that we already discussed. So when there's low blood sugar, you'd have glucagon released from the pancreatic A cells. And the other system would be through the hypothalamic pituitary adrenal axis. And we kind of touched upon this. When there is sort of stress, you'd have a lot of breakdown and increase in glucose level and mobilization of the fuse so that you provide energy for the body. And part of that fuse or part of those fuels would be glucose itself. Now, for the hormones that are involved in these two systems, we already know glucagon is in this system. Now, the other hormones, the epinephrine, the cortisol, the growth hormone, these are other hormones, and we give them the name counter-regulatory hormones because they all counter the actions of insulin. Now, we divided them into two groups, as you can see, a short-term or short-acting hormones and a long-acting hormone. Now, the short-acting hormones would be something like glucagon, and we already know that they increase glycogenolysis and gluconeogenesis. And for epinephrine, this is the other short-acting hormone. It increases glycogenolysis, however, it doesn't increase gluconeogenesis, it increases lipolysis. And it's important for us, these short hormones right now. Now, imagine with me, you have a type 1 diabetic patient, and it's on their later stages. They would have a deficient glucagon release. So if you remember, type 1 diabetic patients, they have an autoimmune reaction in their pancreas, and that mainly affects the beta cells, but as it gets into later stages, it affects everything around it. So alpha cells could also get affected, and that's why they might have a deficient glucagon release, right? So what do you think would happen if they are hypoglycemic? When they are hypoglycemic, you essentially have a compensation from the other short-acting hormone, which is epinephrine. So epinephrine will start to get released and maintain the blood sugar levels back to normal. So subhanAllah, alhamdulillah, we have epinephrine as a backup to glucagon. Now, finally, we're going to speak about the types of hypoglycemia. And so we have three main types, and you could guess how they come just by reading their names. So one is insulin-induced hypoglycemia. What do you think would happen here? So essentially, in insulin-induced hypoglycemia, there's a patient or a person that took too much insulin or had a higher dose of insulin. And so this insulin would essentially lead to the hypoglycemia. And how would you treat him? You'd give them either carbohydrates or you'd administer glucagon directly. Okay. The other type of hypoglycemia would be postprandial hypoglycemia. So what does postprandial mean? Post would mean after. Prandial would be eat or feeding. So after they ate, they would get a hypoglycemia. And this is associated with an exaggerated response of the body to the food. And so it would release way too much insulin than needed. And this could lead to the hypoglycemia. Now, the last thing would be the fasting hypoglycemia. And again, from its name, fasting essentially means yeah, so fasting, and this means that the liver, مثلا, where you have a lot of storages of glycogen and stuff, they are depleted, so they're no longer able to release glucose into the blood because you have no storages, and so this would eventually lead to hypoglycemia. Okay, are all those three clear? Okay, finally, onto the questions. So, an 85-year-old woman with diabetes takes an insulin injection before dinner, but falls asleep in her chair and fails to eat. About 3 hours later, she wakes up feeling shaky, sweaty, and confused. What is the most likely cause of these symptoms? Okay, so the answer here would be a low blood glucose level because the patient, before they ate, they took a dose of insulin. They got injected with insulin, but they didn't eat. So this insulin would lead to hypoglycemia. Yes, indeed, exactly. Okay. A 45-year-old obese male with type 2 diabetes was prescribed insulin therapy, but continued to experience persistent hyperglycemia due to high levels of glucagon. EGLP, or an agonist for glucagon like peptide 1 receptor, was added to the patient therapy to suppress glucagon secretion and improve blood glucose control. What is the primary action of glucagon like peptide 1 receptor agonists? Increase insulin release. Any other answers? Okay, so yeah, exactly. The answer is increase insulin release because we already spoke about how gastrointestinal hormones in general help in increasing insulin release. Okay. What is the primary effect of insulin on adipose tissue? So if you try to think of the actions of insulin, it's an anabolic hormone. So we could start eliminating an answer one by one. So it's anabolic hormone. There would be no lipolysis. Gly glycogenesis, glycogenesis, glycogenesis could be possible, but it's not usually in adipose tissue, but that's also eliminated. Glycogenolysis, it does the opposite, it builds. And gluconeogenesis, there wouldn't be gluconeogenesis, there would be um, glycogenolysis. Um, so the answer would be lipogenesis. So there will be buildup of lipids in the adipose tissue. Does that make sense? Okay. Our last question would be, how do sulfonylureas primarily work to lower blood glucose level in individuals with type 2 diabetes? Excuse me, why is it not C in the previous one? Glycogenolysis? Insulin is an anabolic hormone, so it wouldn't break down glycogen to increase the levels of glucose in the blood. Yeah, glycogen synthesis. So you might ask مثلا why B? Okay, why B is wrong, huh? So B would be wrong. Yeah, it's fine. B would be wrong in this case because it's in adipose tissue itself. So within adipose tissue, it starts building up fat, not glucose. Even if you remember, we already said that one of the actions of insulin is that it stimulates GLUT4 transporters on adipose and muscles. Huh? And these GLUT4 transporters, if we go back a couple of slides, oh. Yeah. So we said it increases glucose uptake in adipose and muscles through GLUT4 transport. And on the lipids, it says that it increases glucose uptake in those in the adipose tissue for triacylglycerol synthesis, or essentially lipogenesis, lipid genesis, synthesis of lipids. Hope it makes sense. Did it make sense? Oh, okay, yeah. Okay, our final question is, how do sulfonylureas primarily work to lower blood glucose levels in individuals with type 2 diabetes? Okay, D. Any other answers? Yeah, you're right. D. So remember when I spoke about sulfonylureas, it's صراحة a very pretty high yield topic. You could get easy asked about like this. And so we said that given to type 2 diabetic patients, and it inhibits the potassium sensitive ATPase. And so by doing that, you're trapping potassium inside the cells. There is depolarization, and calcium would enter. Insulin would fuse, insulin vesicles would fuse with the cell membrane and release the insulin to the blood by stimulating insulin secretion by the pancreas. And we're done, I think. Yeah, thank you. And please do fill this feedback QR code. And thank you for your time.
Thank you.